Copyright Stanford University, all rights reserved.
Inherited genetic disorders in the era of genomic medicine. And the overview of the talk is that we'll be discussing inherited genetic diseases, and especially the move from single gene analysis to genomes. I'll talk to you about some databases and very useful resources. You can use these every day, even if you don't have a lot of knowledge of genomic medicine. And I'll also talk about structural variations in the genome and how those affect inherited genetic conditions. And then we'll go into the identifications of genes associated with disease. And I'll go into examples of how these new methods can be utilized to identify disease genes in methods where we don't have, in diseases where we don't have the genes identified. And I will use two landmark papers, one that identified the gene for Kabuki syndrome by whole exome sequencing, and the other one that used whole genome analysis to identify the, one of the genes in Charcot-Marie-Tooth disease. So first, like I said, back to basics. You do need to know the patterns of inheritance whenever you deal with inherited genetic diseases. So very briefly, just to refresh your memory, with autosomal dominant disease, males and females are affected in equal proportions. You have to have one mutation in order to get the disease. With autosomal recessive inheritance, the parents are typically unaffected carriers. And in rare autosomal recessive disorders, there can be consanguinity in the families. But with cystic fibrosis, for example, that is really uncommon. So with more common conditions that are autosomal recessive, consanguinity does typically not play a role. And such parents have a risk of 1 in 4 of having an affected child, and you have to have 2 mutations to have the disease. With X-linked recessive inheritance, shown here, there's no male-to-male transmission. The females have 2 X's, and they are typically unaffected carriers, and the males are affected. With X-linked dominant disease, again, no male-to-male transmission, but males and females are both affected, and you get a pedigree looking like that. Now, complex pedigree of mitochondrial disease, where you can see that the transmission occurs entirely through the maternal line, but males and females can be affected. And in this pedigree, there's actually the interesting phenomenon of skipping of a generation. So that's a phenomenon called reduced penetrance. So these patterns of inheritance are great when you're thinking about rare genetic disorders. They usually work really well in that context. But, of course, our patients do not read textbooks, and there are exceptions. So if you think about a common recessive trait, then in a pedigree, it can actually really look like a dominant disorder. And a really great example of that is blood group O. Multiple different people can have brought blood group O into a family, and then when you look at the actual pedigree, it can look like it's autosomal dominant. We already talked a little bit about non-penetrance, and when you look at age-related penetrance, and this is an example of Huntington's disease, which is a trinucleotide repeat neurodegenerative disorder, then here curve A, yeah, you can see that better than I on the screen actually. Here is the risk of somebody who has the repeat expansion of manifesting the disease over life. And curve B is the risk of an unaffected child of an affected parent to actually have inherited the disease gene. So age-related penetrance is a very important concept in genetic counseling. Variable expression is something that is seen in autosomal dominant and as well as in autosomal recessive conditions. And it just basically means that in different individuals, there can be different disease severity. And that's a term that comes back over and over again in a variety of different conditions. Then also, you don't get Mendelian inheritance when there are imprinted genes. And that's important in conditions such as Beckwith-Wiedemann, Angelman syndrome, Prader-Willi syndrome, where it is really important who is the parent who actually transmitted the mutation and who had the silenced gene. Male lethality is something that can be seen in X-linked dominant conditions. And what you then see in the pedigree is only affected females. Because, and then you think, oh, you know, this is a disease that only affects females, whereas actually they have two X's and they are more mildly affected, and the males just die prenatally. So that's the phenomenon of male lethality. And then, of course, how can you establish a reliable pattern of inheritance when you have a new mutation or when you have really small families? So that is a real conundrum as well. So lots of complications to the traditional patterns of inheritance. No one ever said it would be easy, right? So in terms of genetic determinants, I really like these two pictures where, you know, even if you have something that is purely genetic, there are multiple different factors that are associated with the expression of disease. There can be reduced penetrance, and multiple genes can play a role. Also, though, if you have a genetic disease, it is influenced by environmental factors. So for any given condition, really, it has its point somewhere within that triangle. And then also multifactorial inheritance is illustrated very nicely here, where we have a scale with all these angels and all these little devils. And so the devils represent the negative factors that contribute to disease. And you can imagine if you add one little devil, you might tip the scale towards disease, but it's not that one little devil that actually is the cause of that disease, right? So there are always major and minor genetic determinants that actually influence the expression of disease together with environmental factors. Okay, so in the diagnostic lab, especially now, we have to contend with lots of different things. So for example, if I take the example of the phenotype of hearing loss, that can be caused by mutations in any of about 200 different genes. So think about it. You know, you have to, in a diagnostic lab, find the cause of the hearing loss in a patient. Well, good luck with that. It's like a needle in a haystack. And that is called locus heterogeneity. Many different genes that can contribute to disease. In a disease like cystic fibrosis, there's only one gene, but there are many different combinations of mutations within that gene. We know about 1,400 different sequence variants in that gene, and you can have, you know, sequence variant A and B, or B and F, or, you know, C and Q, and they can all combine together. It's a recessive disease, so you need two different mutations, but it can be any combination of mutations, and that's called allelic heterogeneity. So the challenge in a diagnostic lab really is that you often have to look at multiple different mutations and at multiple different genes, and how do you do that? It's very important to do that in a logical way, though, because it affects how you make the diagnosis, and that has an influence on prognosis of the patient. It influences how accurate your counseling can be, and of course, ultimately also the management and the treatment of your patient. So the technologies that typically have been used for such approaches are Sanger sequencing, where you really amplify individual exons in a gene, and then you can look at just a few exons or at all of the exons, capturing the coding region of the gene, and then hopefully you'll find a mutation such as shown here. For larger mutation panels, or where you want to look at multiple mutations in multiple genes, you can use small arrays such as arrayed primer extension arrays, that is shown here, or you can use large resequencing arrays, and those are really great for population genetics. And then, of course, there's the promise of the next-gen methods. For many of us, quite a black box, or in this case, a blue box still. But you know, these methods also hold a lot of promise. No matter what you use, though, in a diagnostic laboratory, you have to be able to interpret what you find. And how do we do that today? Well, we use the locus-specific databases. There are such databases for many diseases. We use the SNP databases. Of course, the literature has something been published in multiple patients. We use functional mathematical prediction tools. They're research tools, but they predict whether a change has a functional effect on the protein. And examples of that are SIFT and polyphen. And then we look at conservation across species of the amino acid as well as of the nucleotide. And whenever we can, we do family studies to really see whether the change segregates with the disease. And the American College of Medical Genetics has come up with guidelines for variant scoring. So if a sequence variant has been reported and is a recognized cause of the disorder, then that is category 1. If it's novel and of the type that would be expected to cause disease, and here we're looking at frameshift mutations and premature stop codons, both types of mutations lead to a truncation of the protein, then that's category 2. Category 3 is when they're novel and typically missense mutations, so you don't really know whether they're going to cause disease or not. Category 4 is when they're novel and probably not causative of disease, and these might be mutations further out into introns where there might be an effect on splicing, but it's not very likely. Category 5 is when they have been reported but are actually known to be neutral variants, so that is something you would find in those SNP databases. And then number 6 is something that is not known or expected to be causative disease, but happens to have been found in a patient who has disease. So obviously we're looking at patients who happen to have genetic diseases, and you do find incidental findings in those patients as well. And sometimes if there's only one case in the literature, of course, that mutation all of a sudden is a mutation, so you have to be very critical in how you interpret such case reports. Okay, so I wanted to give you an overview of some really useful databases, good for boards, good for general genetics knowledge, and very accessible. So gene reviews or gene tests, you can see that here, is accessible through NCBI, and it has an up-to-date library of reviews of genes and genetic conditions. Really great study material. There also is a laboratory directory that tells you where testing can be done, a clinic directory, and then other great materials as well. So digging into this database of gene tests a little bit further, when we actually type in CFTR, which is the gene that is associated with CF, then here you can see a tab for testing, where the testing is done, who's doing research. You can tap into that review, other resources, online Mendelian inheritance in man, which is also a very nice database, which used to be in books, actually, really big reams of books, and now it's all online. Locus-specific databases, of course. The quality of those locus-specific databases varies greatly by disorder. So some disorders have great ones and other ones, because the maintenance of such things depends on individuals. This is not something that the government supports necessarily. And then there's the Human Gene Mutation Database out of England, and that's actually a very useful database as well. So here you can click on the conditions that are associated, and then you can go into all of these tabs depending on your interest. So let's just look at this OMIM, Online Mendelian Inheritance in Man. Here you can see, the whole story basically of the CFTR gene and also of the associated clinical conditions. And that's a great comprehensive resource that you can use for boards or for talks. Cystic fibrosis also has a really great locus-specific database, and you can do a graphic search. Here you can see all the exons in the gene, all the mutations and types of mutations that have been found, and then you can click on an individual exon and really go down and examine that. So you see the amino acids, the nucleotide numbering, and you can see the missense mutations. The green dots are deletions, and the pink squares are insertions. And so all the mutations are listed there, and it's a very, very nice resource. If you click on one of those, you get a little description, which is not necessarily complete. You always have to go to the literature, but it is helpful to know whether something has been reported and how it was reported first. Okay, so that's that, and let's now move on to structural variations in the genome. With the advent of these tools that allow genomic analysis, and here I'm talking about SNP arrays and about oligonucleotide array comparative genomic hybridization, as well as actually next generation sequencing with paired ends, we now know that copy number variations are actually very common and include more nucleotides and are more frequent than single nucleotide polymorphisms. And they can be caused by insertions and deletions, duplications, triplications, translocations. Multiple different little changes can actually lead to these copy number changes. And they can cause Mendelian diseases or complex traits. and they can be clinically innocent as well. What we do know now is that they have affected human evolution, and they contribute to the genetic diversity between individuals, and also traits as well as the susceptibility of traits. And because they affect the higher architecture features of the genome, they've also been dubbed genomic disorders. So how do we detect these? Well, conventional microscopy, of course, can look at these numeric changes such as trisomy 21. And that's really, that's really not something that is particularly new. But when we want to look at submicroscopic rearrangements, which are typically 5 megabases or smaller, then we need to go locus specific. And some of these methods are cytogenetic based, and some of these are molecular. So you're all familiar with FISH, but maybe not necessarily with pulsed field gel electrophoresis, and with multiplex ligation-dependent probe amplification, which detects deletions and duplications in genes, and those cannot be picked up by regular sequencing because the primers don't sit down. Other methods that are not locus-specific that I already touched on are array CGH and SNP arrays, and with next-generation methods, the advantage there is that you can basically use read number as a surrogate for copy number. So it's an indirect measurement, but it is very helpful, and you can use these methods as well. Okay, so copy number variations, especially the small ones up to 50 kb, have been very much under-ascertained, and now are thought to account for about 13% of the genome. Of course, we don't know the total number, all of their positions, the size, and how they are distributed within populations and between populations. And so they raise more questions than answers at this time, but that just means that there's more to discover. They can be inherited, or they can be found sporadically, and they are cataloged in public databases. There's the Toronto database for genome variants, and then there are 2 databases that actually catalog those that are disease-causing or associated with disease. And that's Decipher, which uses ensemble data, and the European Cytogenetics Association database. And both of those try to catalog the ones that are clinically important. Okay, so how do these copy number variations actually lead to phenotypes, right? Well, first of all, they can affect genes that are sensitive to dosage. They can have a position effect on regulatory elements. If you have a seemingly balanced translocation, they can still affect long range the regulatory elements. Also, if you have a deletion, then all of a sudden you can unmask what normally would be a recessive mutation or a functional polymorphism. And of course, they can directly disrupt the coding sequence as well. And here you can see a whole list. So these are the disorders, and here you can see how the copy number variation caused the disorder. But apart from this short laundry list of things, we also now have recognized that they do contribute to complex traits, to disease susceptibility and mental retardation. And in that sense, this is an illustration of an ideogram of human chromosomes. And what they depict here is a selection of loci that are associated with mental retardation. And the red ones are duplication CNVs, and the blue ones are deletion CNVs. So just an illustration. This, of course, evolves all the time, but it's quite impressive, I think, in terms of the number of CNVs that are already known to be associated with mental retardation. Okay, now on to the next chapter, and that is the traditional identification of disease genes. So this was kind of before 1980 even, little history lesson here, where they used position-independent methods. And why did they do that? Well, they just didn't have mapping information, and they did not have the tools to generate accurate mapping information. So what they had to do is really look at the biochemical features of diseases. And once they knew something about that, then they could isolate and characterize part of a gene and then hopefully find the gene. And that worked for a handful of conditions, for thalassemia, for PKU, for hemophilia A, but they didn't know anything about the position of the gene when they started, and that was really not working all too well. And then in the 1980s, positional cloning became available, where they were actually closely mapping multiple affected families in order to positionally find the disease gene. And that worked for cystic fibrosis, for Huntington's disease, for breast cancer, for adult polycystic kidney disease, and many other conditions. And I'm using Duchenne muscular dystrophy just as an example. No biochemical basis of the disease was known at all. And then in the early 80s, several groups started to compete to find the gene. And it wasn't until 1986 that both groups almost simultaneously, through different breakthroughs, found the gene. One was a chromosomal deletion in the patient, and the other patient had a translocation. And that's how they were actually finding the dystrophin gene, and or the DMD gene that encodes dystrophin. And it's important to realize, I think, that without having found the gene, we would still not know how dystrophin is anchored in the sarcolemma and how the the contractual or the contracting machinery of the muscle cells actually works. So finding that gene was really pivotal in Duchenne muscular dystrophy. But positional cloning can be desperately hard work. It is just really not straightforward, and there are many roadblocks. And one researcher has put this in a very telling graph, I think. So you can have phenocopies, you can have contigs that aren't perfect, you can chase the wrong gene, you can find benign polymorphisms, you can get a major headache out of all of this. And so by 1995, it was really disappointing that despite all of this very hard work, only 50 genes had been identified by positional cloning. And once you have a candidate gene, you also have to still confirm it. I have to say that now with the Human Genome Project, which started after that, of course, there's much more information, and it has sped up the project and the process. But still, there's some work to do. But when you have a candidate gene, finally, after all that hard work, you have to confirm that it is actually the right gene. So the most straightforward way to go about that is to basically sequence affected individuals and to see whether there are mutations in that gene, and they have to be unrelated individuals. So that's fairly straightforward. You can also restore the normal phenotype in vitro. For example, you can transfect the cloned normal allele into your cultured disease cells and do it that way, or you can make a mouse model of disease, and the transgenic mice have been extremely important in that. So if you have a gene where you're suspecting a loss of function, then you can do a knockout of that gene in the mouse and see what happens. Or if you suspect a gain of function, then you can put in knock in, basically, the disease allele. And so all of that leads to better understanding of gene function. So now to next generation methods. Again, it's a bit of a mystery box for some, but hopefully I'll show you that there are some goodies to be found in that area as well. And it has a lot of promise. So we'll go into two different key papers now. And the aim of the first paper was to discover the gene for autosomal dominant Kabuki syndrome by exome sequencing. Now, why hadn't they found the gene after it had been discovered in 1981 in Japan? The reason was that it's rare, that there were no multiplex families, and that there was a lot of variation, phenotypic variation. So it just wasn't straightforward to pursue this. But you might wonder, what is Kabuki syndrome? So it's called Kabuki syndrome because it was discovered in Japan, and the facial features of these kids actually resemble the makeup that is used by the Kabuki theater artists in their art form. And originally it was called Kabuki makeup syndrome, and then they dropped the term makeup. But the facial features that you can see in these kids are these high arched eyebrows, very long eyelids with dense what's the word? Eyelashes, thank you. Eyelashes. They also have a flat tip of the nose and prominent ears. And worldwide, only 400 cases have been described. The incidence listed here is the incidence in Japan, but all ethnicities have been affected. And so it's fairly rare. rare. We think that it's autosomal dominant because there have been about half a dozen cases where the parents actually transmitted the disease to their child, but most of them are sporadic. They also have mild to moderate mental retardation. They have irregularly shaped vertebrae. They have short fingers with some webbing. They can have scoliosis. They're hypotonic, and they can also have joint laxity. And then the cardiac anomalies include coarctation of the aorta as well as atrial and ventricular septal defects. So there are quite a few phenotypes associated with this. So this group set out to do exome sequencing. At first, they did an enrichment step in which they used hybridization of shotgun fragment libraries on custom arrays. And you can see that process here, where the red bars are the ones that are not desired, and the blue ones are the ones that are desired. They hybridize and they are eluted. And then they used the Illumina genetic or the Illumina genome analyzer, where you basically do your isothermal bridge amplification on the flow cell itself. So you don't do an emulsion PCR before, and that's a feature of several of these other platforms, but not Illumina. So you do that and you get a clonal amplification, and then these are sequenced. You come in then with your additional nucleotides and your polymerase. There's one incorporated, and you image the array because then you have your fluorescent picture. Then you remove the label and the terminator, and you can do that whole process again. So that's all in a nutshell. I know that this was covered in another lecture. So this group worked under the assumption of a dominant model, and they had exomes of 10 unrelated patients. 7 were European, 2 were Hispanic, and there was 1 of mixed European Haitian descent. And they focused, of course, on rare variants because, this was not expected to be something common. So they focused on changes of one amino acid to another, splice mutations, insertions and deletions, and novel changes. And after this very stringent analysis, they only found 1 gene that was shared across the 10 exomes. And they thought, bummer, this is a false positive. And the reason that they thought that was that it's a very large gene and it wasn't likely associated with Kabuki syndrome. Now what, right? They actually thought, well, maybe it is genetically heterogeneous, and the 10 patients that we have don't all have mutations in the same gene, or the mutations are just not located in the targeted exome, or couldn't find them there. And they could also be in between genes or deeply in the introns. So then they resorted to a less stringent analysis. They relaxed their criteria, and they looked for candidate genes shared among subsets of their patient cohort. So they said, if any out of the 10 exons have 1 or more changes, then we're going to categorize that. And how to prioritize that? Well, they did that based on phenotype, and they also looked at the functional impact of the changes that they found. And doing that, they actually found nonsense variants, so premature stop codons, in 6 of their patients, and a frameshift mutation in 1 of their patients, in 1 gene, in the MLL2 gene. And that was very promising. So now they had something identified in 7 out of their 10 patients, and they were really excited about that. Of course, you have to confirm that because, you can have false positives with next-generation sequencing methods, and they did that by Sanger sequencing. And in the remaining 3 cases, they also applied Sanger sequencing to that very gene to see if they had missed something. And they also did array CGH. Well, sure enough, they found frame shifts in that same gene in 2 out of the 3 patients. And so in 9 out of the 10 patients, they now had an explanation for their Kabuki syndrome. And then they screened an additional 43 patients with this syndrome, and they found a mutation detection rate of 60% overall. So it's probably really not that one gene that causes all of the Kabuki phenotype, but that was a really big breakthrough because no gene was known for it before. And what is this gene actually? So it is a gene that is a methyltransferase, and the Drosophila homolog is the best characterized of this set family of proteins, and it is called trithorax. And in this gene, they found several novel mutations, and then two, and those are the underlined ones here, that were actually transmitted from parent to child. And okay, so the next article then was the one about Charcot-Marie-Tooth neuropathy. This is a disease where many different genes are actually known, but it was elusive in this family. They had 4 affected individuals, so 4 affected siblings, 4 unaffected siblings, and the unaffected parents. And they thought, well, nobody else has done this successfully, so let's do whole genome sequencing and see what we can find. And it made sense in this family, as you'll see in just a couple slides. Charcot-Marie-Tooth, as you know, is an inherited peripheral neuropathy. There are 2 forms. There is the form that affects the glia-derived myelin, and then there's the form that affects the nerve axons. And if you have adult onset Charcot-Marie-Tooth, then it's very variable. You have a distal symmetric polyneuropathy and atrophy of the peroneal nerve and progressive muscle weakness, especially distally. So here you can see this horrendously complicated algorithm for the disease, and you can appreciate that there are 3 dominant forms of the disease, 2 recessive forms, and an X-linked form. And actually, out of what is known, there are 39 loci that are thought to be associated with CMT. Out of those 39, genes have been found at only 31, and only 15 of these are actually available for clinical testing. So there's a lot still unknown about this disease. And this family has had quite a good sequential workup. So imagine for a family to go from one gene and to another, to another, to another. So a painful process. And the 5 genes indicated with the blue arrows here are the ones that had already been tested in this family. So some are in this recessive category and some are in the dominant category. And so this is the family that they wanted to study. Now, on this table, you can see the generations. This person actually does have some medium nerve entrapment, but he was a carpenter for 50 years. So that was maybe not directly related to an actual disease. The proband here is in the 3rd generation. This is the age at which these individuals were actually examined. Here you can see their sex and whether there was demyelination. And here's the diagnosis. So Charcot-Marie-Tooth or mild median nerve mononeuropathy, that's MMN, and then some additional information. So they did whole genome sequencing rather than exome targeted sequencing because they thought maybe the mutations are simply not in the coding regions. And they were also worried about missing copy number variations. And the interesting thing is that their study lasted 6 months. And in those 6 months, they had a 3-fold increase in sequence yield without any appreciable increase in cost. So that's how rapidly this technology evolves. And you can see here the read length doubled and the density of sample increased and the technology improved overall. They also did array-based CGH. First, actually, they wanted to compare the proband to a normal male control. And they used 3 different arrays, one from Agilent, one from Nimblegen, and then a 44K array also from Agilent that was focused. It was custom designed to focus on neuropathic disorders. And so they looked at it with 2 approaches, and they found 234 copy number variants from smaller to quite large, and 132 were confirmed by at least one other method, and 94 of them overlapped with reported CNV regions. So there was really nothing exciting there, nothing known to be involved in CMT or other neuropathies. So then they did the next generation sequencing step, and they used another platform. They used the ABI Solid, which is preceded by an emulsion PCR step, and then a ligation-based sequencing. So this is a little less intuitive, I think, but you have a two base encoding method here and a ligation step, and then you have your fluorescent labels as well, and ultimately you derive the sequence just as well as with the other method. And that's what they applied. And by doing that, they found more than 3.4 million SNPs, which is 0.1% of the entire haploid human genome. And those numbers were similar to what had been found in other individuals who had been whole genome sequenced as well. And they found about 500,000 of novel SNPs. Of course, they were most interested in those that could cause disease, and so that could be splice site ones or ones that were non-synonymous, etc. And first, they compared what they found to the Human Gene Mutation Database, one of the resources I mentioned earlier. And 174 were listed there, and 159 of those did not have any question marks associated. So out of those, 21 were associated with Mendelian disease. 16 of those were found to be heterozygous in the patient, and that's actually quite expected. We all carry mutations, and as long as we're just carriers, that is not a problem. So, that was quite expected. But then there were 5 where there was a question of whether these were erroneously called in the database because the patient was homozygous for 4, and then they were also found to be homozygous in completely unaffected individuals. And so then there was one that was heterozygous, and that was a dominant mutation that caused adrenoleukodystrophy, and the patient clearly didn't have that. So, either there is just, that the entry is wrong in the database, or there simply is reduced penetrance for one or more of these 5 changes, and the disease didn't manifest in our patient. So then they focused on the neuropathic conditions, and they examined putative mutations in 40 different genes. They had about 3,000 SNPs. 54 of those were in the coding region, and 2 out of those 54 were at the SH3 TC2 locus. One of them was a premature termination, so leads to a stop codon and probably degradation of the protein, and one was a novel missense mutation. So, that was kind of promising. This gene is a gene that has these SH3 domains. These SH3 domains are important for the assembly of protein complexes that then attach to proline-rich regions, and then there are the TPR domains, and those are important for protein-protein interactions. And so they did segregation analysis in the family, and they thought, we're going to confirm this by very traditional methods, and that's what they did. They amplified and sequenced the affected exons where these 2 mutations were located. And then they also found that this premature termination mutation abandons the restriction site. So that's really, it's a little redundant, because if you already sequence it, then you can pretty much definitively make that call. But they did this extra thing as well. So here you see the pedigree. And interestingly, so when you do this restriction enzyme thing, the larger band indicates that there is a restriction site abandoned when there is this premature stop codon, right? So then you get that larger fragment. But interestingly, everybody who was affected with CMT actually did carry those 2 mutations, indicating autosomal recessive inheritance. You could also see that one of the mutation had come from dad and one of them from mom. And actually, some of these patients had an axonal neuropathy, even though they just carried 1 mutation. So from genotype to phenotype, it seems that the heterozygous mutations can also lead to symptoms, and they hypothesized that haploinsufficiency for this gene may cause mild polyneuropathy, specifically carpal tunnel syndrome. But of course, this could be influenced by other genes as well. You cannot be sure that it's really just that one change. But if you want to tease that out, you would have to do whole genome sequence analysis on multiple different other family members, and I'm not sure that they were prepared to undertake that big endeavor, especially since that was not the major condition in the family. So in conclusion, we have talked about inherited diseases, the patterns of inheritance that we need to look at when we deal with patients, and the things that influence these traditional patterns of inheritance. We talked about the evolving methods of diagnosis, also about databases and resources that you can use. We discussed structural variations and how the diagnosis of those actually evolves, and then the traditional identification of disease genes, as well as the new next generation approaches specifically targeted on whole exome analysis in Kabuki syndrome and on finding the gene by whole genome analysis in Charcot-Marie-Tooth disease. With that, I'll be happy to answer any questions, and thank you for your attention.
Copyright Stanford University, all rights reserved.